Hello everyone, welcome to another IVM's webinar. I'm Kübra from IVM Technology and we are pleased you could join us today. In this session, we will be discussing intravital in vivo imaging for advancing pharmacological research in subcortial brain regions. We will cover the use of cranial imaging windows, imaging cannulas, other brain accessories, and fluorescent labeling agents to target specific structures in the brain, such as blood-brain barrier, immune cells, or blood vessels. While listening to Hayden's presentation, you can find more information about these tools on our website and shop page, ivmtech.com and ivmtech.shop. Now, I'm happy to introduce today's presenter. Some of you may already know her. Hayden is a senior technical applications specialist at IBM, and she has extensive experience working with both in vivo and ex vivo tissue samples, including human brain tissue, and possesses deep knowledge of brain regions and mechanisms. And today she will provide us with a broad overview and share some of the latest developments in brain at brain imaging using intravital microscopy. Before we begin, please feel free to submit your questions in the Q&A box below throughout the presentation, and we will do our best to answer all the questions at the end. Hayden, if you are ready, please go ahead and start with your presentation.
Okay, so could you hear me well?
Yep.
Okay, so hello everyone again. I'm Hayden, an application specialist from IVM Technology. So it's very nice and honored to have you today, today's webinar. So today I'm going to introduce our cutting-edge confocal and two-photon compatible intravital imaging system, highly pivotal for acquiring cellular and pharmacological activities from brain in living animals. So please leave your questions during the webinar if you have any, like you said previously. Okay, so first I'd like to give you a brief overview of intravital microscope. So intravital microscopy, or IVM, is a powerful imaging technique that lets us watch how cells and other molecules behave in real time inside of the living animals, especially the mouse or rat. So, unlikely the traditional methods like histology, for example. So, IVM shows what is happening in a natural and living environment. So, we can actually observe things like how cell moves, interacts, or responds during disease progression. So, compared to traditional and conventional imaging systems like X-ray or bioluminescence modalities, IVM offers something very different from them. So it gives us high resolution real-time images at the cellular level. And also this makes IVM a valuable tool for today's pharmaceutical and biotech researchers where dynamic and detailed real-time insight is more important than ever. So there are already well-known in vivo imaging tools like X-ray, PET, MRI, and uh, other modalities, like I did demonstrated before. However, these techniques only provide rough and low-resolution data because they capture images at the whole body or whole organ levels. So to address these limitations, we have designed more advanced intravital microscope that allows researchers to track and follow the cellular dynamics, observe the drug distribution and therapeutic effects, even in challenging organs like the lung, heart, brain, and all in living animals. So to better understand how our body works, it is very important to look at what's happening inside of the body, right down to the level of individual cells, especially in areas like immune responses, cancer, and other drugs to see how medicines or other cells move and act in the body. So intravitreal microscopy lets us watch these changes and morphologies in real time while they are happening inside the living animals. So especially our system makes it possible to study how drugs travel through the bodies and how they affect cells life in real time. So for example, on the left, I'm showing the results from the sepsis, uh, lung sepsis model where inflammation blocks the blood vessels in the lungs. So on the right, we can see how we track exosomes, anti-cancer drug movements, and the transparent deep cells in the body using our imaging system. And like I mentioned previously, these techniques, the macro scale modalities, only provide rough and low resolution data as they capture images at the whole organ level. So to overcome these limitations, we, like I said, we have very advanced intravitreal imaging modalities, which is called IVM. And the distinctive strength of intravitreal microscopy, especially from IVIM's intravitreal system, lies in its departure from conventional in vivo imaging approaches. So traditional methods require the preparation of separated animals for each time point, inherently introducing substantial inner animal variability. In contrast, our system enables longitudinal imaging within a single living subject at precisely defined time point, dramatically enhancing consistency and reliability. So more than just static image acquisition, it empowers the real-time dynamic visualization of various biological processes as they unfold. So here comes our all-in-one system designed for superb and highly optimized intravitreal imaging performance. So this compact single box package allows for easy installation, operation, and maintenance, which makes it perfect imaging tools for any lab environment. So this system also combined co-optimized hardware and software featuring an ultra-fast polygon scanner speeding up to 50 frames per second, temperature monitoring system, motion compensation, and also the AI-based denoising for stable and high-resolution imaging. And most notably, with this automatic and hassle-free motion compensation, live animal maintenance tools, and other window options, researchers can obtain high-quality in vivo data across a wide range of organs without the usual limitations. So, by using these various types of implantable imaging windows, our system supports longitudinal studies. So, this capability allows for continuous observations of specific tissues over days, weeks, or even months without the need for repetitive surgical interventions for the imaging. So in today's presentation, I'll be showing imaging results from a mouse model equipped with the cranial imaging apparatus, highlighting how different cells and drugs respond dynamically over time. And with this IVM's highly optimized systems for imaging, you would see the immune motilities in various animal models in real time, disease progression longitudinally, and also the drug distribution or efficacy within the same subject over time, rightly in brain as well. So, okay, now that we have covered the introduction, so let's move on to the case studies where we acquired imaging data using the confocal and two-photon capabilities integrated into our IVM microscope with employing various mouse brain disease related models. Okay, so let's take a closer look at brain imaging. So as introduced in the previous slide, IVM technology uses the window implantation technique to enable the brain imaging. So on the left, you can see a 3 mm diameter craniotomy covered with a glass window and fixed with adhesive. So this setup allows direct visualization of the cortical surface, and the method is commonly referred to as a cranial imaging window. However, this approach has its own limitations, which is it allows imaging only within the certain depths of the cortex. So this becomes a challenge when studying diseases like Alzheimer's, Parkinson's, or other things where deeper structures such as the hippocampus are of interest. So to overcome this, we have developed a cannula with a cylindrical imaging window. And by removing a small portion of the cortex, this device enables the objective lens to physically reach and image the deeper brain regions. And this video demonstrates the real-time visualization of cellular activity in the brain using a mouse model implanted with a cranial imaging window. So we used the Thywan GCamp 6F transgenic line to monitor neuronal activity through calcium signaling. So neurons fluoresce as calcium ions flow in and out, allowing us to track their activation in real time. So to visualize the vasculature, we also administered a vascular labeling kit, which stains the blood vessels in red. As the video progresses, you'll notice the increasing GFP signals in neuron cell bodies, reflecting their dynamic responses to calcium fluctuations and highlighting the actively firing neurons. And this time, we have implanted a cranial imaging cannula targeting the hippocampus in the same strain of mouse. So as shown here on the right, this approach enabled us to clearly visualize both the blood vessels in red and the surrounding neurons with high resolution. So, okay, it's time to demonstrate the application results. So first, I will present the example data from imaging studies where we have observed various immune responses and other cellular activities in brain disease models. And okay, so now I'd like to share some brain imaging data from photothrombic ischemia model and walk you through how our equipment can be effectively applied. So this will provide a clearer picture of its capabilities and how it may be relevant to your own research. So in this particular study, we induced the photothrombosis by injecting rose bengal and irradiating the targeted area with a 961 nanometer laser. So using the DHE and PI staining, we were able to visualize the reactive oxygen species and cell death, confirming that photothrombosis was successfully induced. Blood vessels were also labeled and imaged from day 0 to day 30. Following photothrombosis induction, we initially observed the signs of brain swelling, like due to the inflammatory responses, I think. But however, by day 8, the imaging revealed that the brain had actually contracted even more compared to day 0. And in addition, fluorescent dyes of different molecular sizes were injected to assess vascular permeability. We observed that the smaller 3 kilodalton TriTC dye leaked more rapidly across the blood-brain barrier compared to the larger 2 megadalton FITC dextran dye. So this finding suggests that the BBB was likely compromised following photothrombosis and microinfarction as well, resulting in vascular leakage. Interestingly, by day 5, the leakage of TriTC had returned to baseline levels. So you can see the same result data on the right, indicating that the partial recovery of BBB integrity. So through this investigation, we aim to identify which cell types contribute to the recovery of vascular leakage around the 3 to 5 day time point. So our results show that NG2 positive pericytes, which is expressing the red color here, underwent division and elongation along the vascular walls following a microinfarction. So this finding suggests that pericytes play a direct role in the restoration of blood-brain barrier integrity. So building on our previous data showing that pericytes are involved in the mechanisms of blood-brain barrier damage and recovery in a brain infarction models, so we next examine the responses of various immune cells involved in inflammation as well. So as you can see here, photothrombic ischemia and infarction was induced in the same way by injecting a Bengal followed by irradiation with a 961 nanometer laser. We then injected the labeling kit based on CD31 antibodies conjugated with the far red fluorescent dye to mark the blood vessels in red here. So compared to the control model results, the infarction model exhibited more irregular and disrupted vasculature. And even when examining the multiple regions, we constantly observed a significant increase in the number of immune cells labeled in blue, indicating an active inflammatory responses. So in this slide, now you're seeing the red colored blood vessels and also the blue colored monocytes, which is expressing the TD tomato. And by counting and comparing the number of these immune cells, we found approximately an 8-fold increase following infarction. So this supports our explanation that brain tissue likely swells due to the inflammatory response after infarction. So importantly, we were able to directly observe these swelling responses in real time and also the related immune responses as well. And similarly, after implanting the cranial imaging cannula, we investigated whether the same immune responses occurred in the deeper hippocampus compared to the cortex. So by imaging the same location before induction and then at 1 hour, 6 hour, and 24 hours post-induction, we observed consistent patterns of cellular responses, especially the neutrophils. So this demonstrates that with our IVM technologies, two different surgical techniques combined with intravital microscopy imaging system, it is possible to repeatedly visualize the dynamic cellular responses multiple times within a single animal. Therefore, this platform serves as a powerful tool for understanding the disease onset and progression mechanisms. And this time, I'll present example data of long-term observations of various cellular responses in brains following drug treatment related to the disease. Okay, so here, a cranial imaging window was implanted in a transgenic mouse expressing GFB upon activation of calcium-liberated channels triggered by calcium ion influx. We then stimulated the mouse using von Frey-Flemeth to activate these calcium channels. So following this, we also administered pain modulating drugs such as aspirin and performed the real-time cortical imaging while the mouse was awake to observe the cellular changes in terms of the activation of the channel. So in these two videos currently playing right now, you can clearly see that the activation of the calcium channels in the mouse on the left after aspirin treatment is significantly reduced compared to before treatment mouse. Since the mice under anesthesia do not perceive pain, stimulation with one prey filament fail to trigger any detectable calcium channel activation. However, awake imaging allows us to clearly observe this activation. So to facilitate this, IVM technology offers a range of accessories and various adaptable tools designed to support imaging in both anesthetized and awake states, helping researchers perform experiments more effectively. And another mouse model was implanted with a cranial imaging window in a transgenic mouse I'm sorry, uh, we have implanted the GFP expressing brain tumor cells, which is called GL261 GFP cell lines, to the mouse brain directly, and also the cranial imaging window implantation was followed after that. So before administering the nanoparticle anti-cancer drug, we initially tried to see the successful growth of the cancer cells in mouse brain. So after 7 days, we could successfully find the tumor cell insection site, and also with the right wide area imaging of the orthotopic in orthotopic mouse models at 7 days, we could also see and observe the successful full growth of the tumor cells as we intended. So we have monitored the cellular responses from day 1 to day 14, and I have employed only the 4 data from day 1 to day 4. So in these images, you can see the red color nanoparticles are accumulated very close to the grown tumor cells in the brain at day 1 point. And after day 4, and after 4 days after drug administration, you can see the red color nanoparticles slightly infiltrated and accumulated in the brain tumor tissue, and also the infiltration of the immune cells, which is now expressing the white colors, are also successfully observed from day 2 to day 4. So this demonstrates that IVM equipment enables not only the observation of simple cellular responses, but also the real-time monitoring of diverse drug effects within a single animal, providing consistent and reliable data. Okay, so that's the wrap-up of my brief introduction of our system and also the reference data acquired during the our internal studies. So let's move on to the conclusion. Okay, so intravital microscopy as well as the IVM has transformed biomedical research by allowing real-time, high-resolution imaging of cellular activity in live animals. Also, the IVM offers key insights into the drug delivery, immune responses, and other long-term disease progression. So as technology evolves, IVM will remain essential for understanding complex biology and developing better treatment for human diseases. So to follow these trends, we also provide the optimized in vivo imaging solutions with transplantable imaging window chambers, accessory fixtures, mounting parts, also holders optimized for high quality observation, followed with the animal body temperature monitoring and maintenance system. So with IBM's intravital microscope, you could successfully achieve high quality live cell imaging in nearly all internal organs in live mouse models. And also beyond the mice, our product line supports imaging from the small scale animal models, the mouse for example, and to the medium to large scale animals rats, ferrets, guinea pigs, rabbits, for example. And also we could provide some robotic arm system for non-human primate animal imaging. So this is the customer portfolio of ours. So we have various global partners and key buyers in and the domestic customers as well. So thank you for your attention. And if you have any questions, please leave your questions at the Q&A box. Thank you.
Thank you, Hayden, for the excellent presentation. As Hayden said, if you have any questions, please feel free to share them through the Q&A box.
Okay, I have received a question. So one of the attendees asked me how long and how difficult to implant these two different types of imaging window in a living animals. So with various triple or twice or triple personal training, maybe it takes about 30 minutes to 1 hour to implant the imaging windows and the imaging cannula in the living body. So, and also it maintains about over a year and a half. So this is very powerful tool for studying various brain-related diseases, as you shown in the previous slide. So if you are curious about the on the surgical protocols, maybe we could provide you and follow you up with the surgical videos and some good images for you. And also we are providing the training services as well. So maybe if you have any inquiries, please leave comments on the survey to follow you up. Thank you.
Okay, we have a question about the JP model in your slides. What was the route of tumor cell injection for that study?
Okay. We just raised the cell the GL261 GFP cells in vitro condition, and then we injected after mounting the mouse on the stereotactic stage, which the mouse when after the mouse is under anesthesia, and also with the Hamilton syringe, we just slightly injected the tumor cells in the cortex tissue.
Mm-hmm. Another question is about working distance of the objective lens.
Mm-hmm. We usually recommend using the 25 water emulsion lens because it performs the best in terms of the confocal and two-photon imaging. And in that case, the working distance of the objective lens, maybe it could be around 1.5, I think. And after this seminar is over, maybe I could give you more precise, detailed information on this via email.
Mm-hmm.
And also the light source is the laser, confocal and two-photon laser in the same equipment.
Yeah. And another question is how to analyze the data regarding the light intensity. He is probably talking about the fluorescent intensity changes or cell immigration.
Mm. So with this, if you want to measure the fluorescent intensity, very simple. We provide our post-processing software, which is called the IVM Studio, is also providing the simple measurement of the fluorescent intensity from each pixels. So in that case, you can compare the GFP, RFP, and Sci5 intensities in that software. But if you like measure to get very exact and detailed data to build some graphs or something else, maybe you could use a third-party software program which Imaris, RVS, ImageJ, or other things, for example. So in that case, you can measure the intensity, but also you can track the immune cell motile immune cells as well. Mm-hmm. In that case, you can also measure the velocity and draw the route.
Hayden, do you also want to talk about the imaging depth for confocal and two-photon modes and for in vivo and ex vivo tissues? This is one of the
Uh,
common questions, isn't it?
okay, yeah, sure. So, for example, the red color organs, liver, pancreas, kidney, for example, they have many erythrocytes in themselves. So, in that case, the imaging depth is not that deep. So, for instance, confocal may go into around 150 or 200 microns, and with the two-photon laser, you can acquire the data with around 200 micron imaging depth. But uh, unlikely, brain is very good tissue for deep imaging because brain, as you know, brain is very pink and they have no blue, red colored cells in it. So in that case, if you like to see the brain tissue with our system, the confocal laser may go into around 300 to 400 microns and two photon around 700 to 1000 microns. And also our lasers, confocal and two photon lasers integrated in our IVM series imaging system, we equip the exact same models from other conventional confocal and other two photon microscopes which you use in your lab. So maybe it's very easy to use.
Mhm. And do you also want to talk about the fluorescent dyes or labeling agents that we
Mhm.
are using for
Mhm.
channels, the wavelength?
So we have eight different detectors. So four for confocal and other four for two photon. So we could visualize the TAPI, GFP, RFP, Psi5, and also the second harmonic and GFP, RFP, and Psi5 with the confocal. So like I said, our system is based on detection of the fluorescent signals. So which means you need to label the target cells, or in other case, you need to employ the transgenic mice expressing the endogenous fluorescent signals. But this is not that easy, as you know. So we also provide the in vivo labeling kits, which could target your own cells to label them with the different colors in your mouse model. So we have various uh, products to label the from like endothelial cells, from the immune cells, the macrophages, neutrophils, and other cells, for example. So maybe with our system and also with our dead labeling kits, you could successfully and very easily visualize your animal models to see the mechanisms or other cellular activities in your to for your research.
Thank you for the answer. And if
Mhm.
there are no more questions, we can wrap up for today, and that concludes our Q&A session. Thank you all for your active participation. And as we mentioned earlier, we have recently launched the IVM shop, where you can find the imaging accessories, fluorescent dyes, fluorescent labeling agents, and also training programs. And by registering an account on IVM shop, you can access certain benefits applicable to your each purchase. For any questions or inquiries, please contact us at information@ivmtech.com. Thanks again for joining us, and we look forward to seeing you at our next webinar.